system. Here we go. I'm using this to start rather than the one because I can get through most of this first. This first PowerPoint is mostly sort of busy work stuff. The two major systems for control, for very rapid controls all the time, nervous system. For longer term controls, acting onset slowly, disappearing slowly, endocrine system. So nervous system is really about, for the most part, rapidity. There's aspects of it, as you will see, when we do the autonomic nervous system, not which have a little bit longer term effects. But in general, rapid and specific, effectively, it's an electrical system that communicates by chemicals. The chemicals, as many of you know, are called neurotransmitters. So effectively, you have an electrical signal, and to transfer that signal to the next nerve or to the next structure, just like the neuromuscular junction, we release chemicals. That, ladies and gentlemen, is the rate determining step. That's what limits the time. How rapid the system is at that. And really, that's the controlling aspect. So we spend a fair amount of time with neurotransmitters, but not right away. This is more about the anatomy today, the microanatomy, if you will, and then some of the mechanics and physiology electrically, and then we're going to deal with the neurotransmitters. That's the last part. So, and effectively, it's very straightforward. You've seen this before. Effectively, you have incoming signals, processing of it, and effecting or having a response to it. This is the minimum of any nervous circuit. We saw that in the peripheral nervous system. Like a simple reflex, you have a sensor, you have a respond, and so you have at least one we call interneuron or association neuron. The vast majority of your nerves are in this category, integration. The vast majority, 99%. So even though you have a ton of sensors and a ton of motors, imagine how much processing goes on. So that's all they're showing you here, nothing major. This is, you've seen this construct already. The central nervous system we did previously, brain and spinal cord, peripheral nervous system, everything that protrudes, that's fine too. We did spinal and cranial nerves. So fortunately, we had a lot of this in anatomy. It's there, not going to go do that. We're going to do autonomic nervous system, so I don't have to say a whole lot about that. The big difference when it comes to somatic, muscles and joints, skin, visceral organs, whether it's sensory, afferent, or incoming, versus motor, efferent, or outgoing. Hence the term effectors from efferent. So you're going to hear a lot going forward about afferent and efferent limbs. You might remember from kidney where we talked about afferent and efferent arterioles and the anatomy of the glomerulus. So these terms repeat themselves a great deal. And somatic, autonomic. We have a whole chapter on autonomic. Very interesting system that really is the heartbeat of homeostasis. What's unique about the nervous system, you do a chapter early on in anatomy that we did in the lab about connective tissues. Remember, we had epithelial tissues, we had lining tissues and connective tissues. None of those appear here in the central nervous system. Okay, and to some extent, even in the peripheral nervous system, it has a unique kind of supporting cast. The nerves themselves, the neurons, we're going to look at. But first, you have to understand what they call glial tissue. GLI literally means glue. So this is neuroglia, as they're referred to, are the glial cells. These are the ones that can actively still undergo mitosis. Remember, nerves themselves, the true neurons, do not. So neurons, when they're damaged in and around the cell body, quote unquote, the nucleus of that nerve, even though it's not the term we would use, cell body or soma is, that nerve is going to be gone. If it happens along the axon or some transmitting or receiving end that had a long thing, yes, we could estimate there's potential for repair. The ones that undergo that's repair, not necessarily mitosis per se. The ones that are able to undergo mitosis, and because of that, when you hear about a brain tumor, this is a glioma or a gliosarcoma, because they're a connective tissue. Brain tumors don't affect the neurons, they affect the supporting cast. You'll see some of the names, and you may have heard about tumors here. So the neuroglia, these glial cells surround, wrap, support, nourish, protect, pick a word, okay, and the nerve cells themselves. You have six main types of cells. Four of these are in the CNS. There are two in the PNS, the peripheral nervous system. The two in the PNS, or peripheral nervous system, are similar in function but slightly different in appearance than the two that they mate with, so to speak. You're going to see something called a satellite cell. When we look at the peripheral nervous system in this slide sequence shortly, they kind of, we think, notice the way I phrase that, do the job of astrocytes, who are by far and away the ones that are the most abundant of the glial cells and have the widest things that they do, sort of scope of activity, and the oligodendrocytes, which are responsible for making the very famous myelin sheath of white matter. All the white matter is ensheathed in a protein and fatty substance called myelin. And oligodendrocytes make this in the central nervous system. In the peripheral nervous system, they are called Schwann cells. So, I mean, people are hearing about schwannomas, not unusual. So we hear about astrocytomas, very famously, typically in the optic nerve area. We hear about a generic term called an optic nerve glioma, because it involves perhaps one, but perhaps more than one different types. These are all relatively common terminology that you'll encounter. So we will go on. The astrocytes. These are pivotal. They play an important role in the blood-brain barrier. They play an important role in growth and development. They play a vital role in nourishment. Okay, all of these things that are there. And what happens, not only do they surround the nerve, they also are there at the synapse, the junction points between nerve and nerve. Okay, and they also surround the blood vessels. There's no direct contact between your blood supply and your brain. In order for material to get from the capillaries, where nutrients and oxygen exit and waste products and carbon dioxide enter, okay, into the bloodstream, they have to go in by way of the astrocytes, as you will see. So it's very interesting, and that's why, and that's the blood-brain barrier is a composition of the capillaries, which have an abundance, okay, of tight junctions. Again, that's the pivotal piece of the blood-brain barrier, is all those tight junctions. And then on top of that, it's surrounded by a glial cell called an astrocyte. So you have effectively extraordinarily large amounts of these tight junctions, not allowing water or water-soluble materials to pass. And then on top of that, it's encased in an additional layer of membrane, the astrocyte. So materials have to be membrane compatible, be able to be transported through the membrane, fat soluble, if you will, small and lipid soluble, oxygen, carbon dioxide, all those things. And that plays a role in medications that we use that have activity in the brain. They all have to be in that category. So there's a fair amount of pharmacology associated with this as well. So they support and embrace nerves. Okay, they play a physically supportive role. The exchange, we'll revisit it shortly. Development, controlling the chemical environment is part of the exchange and the materials that surround it. Yes, they respond sometimes to impulses of neurotransmitters. They play a role, not necessarily well defined, in functioning or in information processing. All of this is there. So they play a really pivotal role. It's an interesting illustration. The yellow is the nerve, the neuron. This would be the cell body, but you can see it's all over. And here is the capillary. So effectively, if something's going to leave the capillary, it has to go through this membrane. It just doesn't leak out again. There's no little openings for water, not appreciable. An abundance of tight junctions. It has to go through the membrane of the astrocyte and then into the astrocyte and then along through that, let's say in here and then out there. So it's traveling along or within the membrane of the astrocyte into, indirectly into the nerve itself. Interesting. And that it gets its name because it's star shaped. You have a lot of these tentacle like projections, so to speak. And so hence the astrocyte, star shaped cell. If I said Astro the dog and the Jetsons, would that ring a bell? Probably. You see George Jetson, his boy Elroy.
Yeah.
See, they had to create a space age family after who? Who came before the Jetsons? Flintstones. Yabba dabba doo.
Didn't like the Jetsons take place in like the 90s?
Probably.
Yeah, it was like that.
Yeah, like we
No,
blown
George Jetson wasn't born until 2022.
Oh.
Chapro. Flintstones were great. I give up. You're one of us. So, microglia. These are sort of like scavenger white blood cells, but not. So, again, the immune. Basically, this is like an immune favored area. So, what this is, these play a role toward injured and debris and phagocytize if there are microorganisms or neuronal debris. So they play kind of a scavenger like cleanup role. That's the best way I can describe it for now. This is what they look like, their rendition. White blood cells look like that as well. A lot of times they have these tentacle like projections, and a lot of those are areas that could generate phagocytosis. Interesting cell. If you recall, when we looked at the central nervous system, we found ependymal cells in and around the ventricles. In the ventricles, you might remember the term choroid plexes. They were capillary beds that were surrounded by cells, and that's where sort of we generated cerebrospinal fluid. Cerebrospinal fluid is not plasma. It's similar to plasma. It comes out of the capillaries. There's modifications in the electrolyte, and the ependymal cells cover the capillaries. They have cilia and they move, so they create a circulation to circulate cerebrospinal fluid. So we find them particularly in those ventricle cavities, lateral ventricles, if you remember those, associated with the cerebral lobes, the fourth ventricle between the third ventricle first, and the diencephalon, and the fourth ventricle sort of anterior to the cerebellum and posterior to the pons, and to some extent the medulla oblongata. So they're a permeable barrier between cerebrospinal fluid and cavities and tissue fluid bathing the central nervous system cells. So when you say permeable, there's a certain degree of exchange that goes on that's there. Again, cerebrospinal fluid is very interesting. It has a lot of similar properties to plasma, and that's one of the things when we do a lumbar puncture, there's a lot, you get a tremendous amount of information, not just about somebody having meningitis, but about other things as well. And this is their rendition of an ependymal cell. You can see the ciliated protections. Here's wherever we're looking at the tissue itself. They're not showing the capillaries per se, but these ependymal cells are creating a flow of circulation. And these are the Schwann cells. And there's a unique difference between, these are the oligodendrocytes, between them and the Schwann cells. The brain, you have billions, if not trillions of nerves in a relatively small space. We can't individually wrap these, if you want to think of it that way. So the myelin sheaths surround multiple nerves. So here this is just showing you 3. They can wrap up to about 60 of them. And here would be a typical oligodendrocyte. You can see, just for argument's sake, it could be up to 60, as I said, or so. Here you have part of the membrane wrapping around one axon, another axon, and another. So what it does is utilizing a limited amount of cells, a lot of cell membrane, and basically compacting all those axons. Where we have, think peripherally, like in your extremities, you got a lot of room in there. You have some muscle and you have blood vessels, but I mean that those big nerves like the sciatic nerve, the radial nerve, musculocutaneous nerve, all those wonderful nerves that we talked about previously, and they're all myelinated. you're going to see a different way of wrapping them. That's there. And so this is, they call these now myelin sheath gaps. Okay, that's the official name because they're gaps in the myelin. And what we learn about in this chapter electrically is how the signal jumps from point A to point B. It's got a famous name that we'll go over. It's called saltatory conduction. It means it's jumping from place to place. It really doesn't jump. It's just the way it's represented so people would understand it. So the signal is from here, and people would think, oh, it jumped from this opening, this space, this gap, to this gap, to this gap. The reality is it's going along the membrane inside, but the myelin acts as an insulator to make it less complex. And the only place where you have all those wonderful sodium and potassium voltage gated channels you're so happy with me telling you about are in abundance here at the gaps. Years ago, they had a different name, the one I learned, and I love to say. It's got French. The sheath gaps are called, are you ready? Nodes of Ranvier. Or if you're Pittsburgh East Grandview. North Versailles versus North Versailles. So you have, right? Nodes of Ranvier. Dems de nodes. The distinction in the peripheral nervous system, satellite cells around the cell bodies in the peripheral nervous system, similar function. They're not very specific. Years ago, function was unclear. And Schwann cells, another name, neurolemma, which is a membrane, neurolemmacytes, surround peripheral nerves and form the myelin sheath in the thicker nerve. And really, if we have a named nerve, it's going to be myelinated. Okay, vital to regeneration and peripheral nerve injuries. That's the one where we see deep cuts, maybe even part of a limb severed off that they can reimplant. That is all because of that myelin sheath. And what's interesting about it, take a look. Here's a satellite cell. This would be a sensory nerve. That would be like a dorsal root ganglion. And so in comes a sensory gap for my finger, and you can see they're individually, it's sort of along that nerve, that particular axon, and there'd be many of these. You remember from the illustrations of the peripheral nervous system, and here are the sheath gaps. Okay, and so effectively they sort of surround it that way. If you go back, the nuclei are here are offset. Okay, but because in this one, as you will see, because each of these is a Schwann cell, the nucleus is kind of in there. So it's a little different. The way they describe it is sort of wrapped jelly roll style. So it's almost like you had a piece of material here and the nucleus here, and you sort of wrapped it around and around and around. So you have multiple layers of myelin. Basically, it's the cell membrane. Because what are membranes? Fats and proteins. What's myelin? Fats and protein. And myelinization is super important. The whole idea of myelinization and the dreaded disease that we see, principally, I told you about my first wife and I being tested for that. They actually had a blood test in the early days of genetic testing. Tay-Sachs disease is a lysosomal storage disorder, but what happens is the lysosomes are, because they can't get rid of the debris, it causes the Schwann cell, it causes the myelin cells to deteriorate. So it's a demyelinating disease. That's how it works. And so eventually the nerves begin to deteriorate. Roughly by age 1, those byproducts accumulate. By age 2, you're seeing the evidence. You're fortunate the baby, or unfortunate the baby survives till age 5. Ain't good. Okay, so you have to know all 6 of those. 2 for peripheral, Schwann cells, satellite cells, oligodendrocytes, astrocytes, microglia, and ependymal cells, the ones in the central nervous system. Know their roles. So nerve cells, structural units of nervous system, neurons, very specialized. You got your whole complement when they're born. As far as we know, there is some information that suggests there are some ones that have limited mitotic abilities. I don't have enough information to share that, or I would never ask you about it. They have to, and again, and when it comes to your brain, only glucose will do. And you have to have oxygen. Without those, big trouble. And so they have what we call a cell body, which you and I might call the nucleus of the cell. But if you remember from anatomy, we use the term nucleus for clusters of cell bodies. The red nucleus, right? Whatever, the geniculate nucleus. There's lots of different nuclei that we use. All the nuclei are the cranial nerves. Those are all clusters of cell bodies. So we use cell body or soma or even the term perikaryon around the karyon and around the kernel. So perikaryon or soma or cell body, and that does everything. So that's where everything's made: proteins, membranes, neurotransmitters. Typically, nerve cells have a certain amount of pigmentation in the rough endoplasmic reticulum. The fancy name that we should call some of these, that's pronounced nacelle bodies, it's a proper name. We look for that. A lot of times when we see irregular pigmentation, that might be an early sign if we're doing it, particularly, wrong way to phrase it. Post-mortem, we would see, in the early days when you could only diagnose Alzheimer's post-mortem, you would see pigmentation deposits, you'd see tangles, neurofibrillary tangles, and neuritic plaques. Those were the three consistent findings. Thankfully, now we can diagnose this earlier with a variety of imaging techniques as well as other things that we can utilize. A lot of it's questions, and unfortunately, the onset of early onset dementia, particularly troubling in a lot of individuals. He's dead now. I did not know them, but my sister and her husband had like 4 or 5 fast sets of friends because they were a relatively new couple when they moved to Rochester. And there's a couple they became very, very friendly with, and this guy was just the life of the party, dentist or an oral surgeon. He was a really good guy, very, very nice fellow. And I mean, and I don't know what occasioned him, but probably in his early 60s, he went to see his family doctor about something, and something triggered it to the family doctor. And he asked him a series of questions. He goes, I want you to know, did you have patients early today? He goes, yeah. He says, those are the last patients you're ever going to treat. He was already in his 60s, starting to have signs of dementia. And I don't know if that caused it. There's one that's pretty well known called Lewy body dementia. Have you heard of that? Yeah, not a good thing. Not a good thing. Because that's unusual. And interestingly, my sister, I don't know if he's still alive. I have to talk to my sister. Her sister-in-law, my brother-in-law's younger sister, about 2 years older than I am, wonderful guy, but for the last 10 years, steady deterioration now. And I think he's now and can't even take care of him now. It's hospice care. Good. If you get older, that whole count your blessings thing, it's a good thing. So the membrane is really where it all shakes. The membrane transmits the signal. Receptors are on the membrane. The transmitting areas where the neurotransmitters are released are on or part of an extension of the membrane and protein appendages. So nuclei versus ganglia, we've talked about this before. That's just a naming contraction. And so we call these effectively these arm-like processes, okay? So peripheral nervous system is mostly these axons. In the central nervous system, we call them tracts. We talked about that a little bit in the anatomy that are there, and they're either dendrites, receivers, or axons, transmitters, to simplify. Classic. This would be a peripheral nerve. Let's say this was in the spinal cord, for argument's sake. Maybe this is a portion of the sciatic nerve. So you have standard, and what you're going to see is you'll have hundreds, if not thousands, of inputs and hundreds, if not thousands, of outputs. It's extraordinary. That's how we process information. Information coming in, either creating a signal, modifying a signal, making a signal stronger or weaker, and then we can convey that to a whole bunch of other things when we're processing information. I'm looking at you and I'm trying to gauge whether what I just said was funny or whether you're paying attention or whether I lost you, which I do most of the time. Okay, all the time. that type of stuff. You get the idea, and you're processing all of those things and making all these changes. It's brilliant how the mind works. It's so fascinating to be able to see that, particularly great minds. So hundreds, and frankly, there's more. They are again. They look like like branches of a tree where all the flowers have come off, all the leaves have come off. Okay, diffusely branched. Organelles are not a whole lot different. You have mitochondria because you're going to have to process and send a signal that are there, energy dependent, and these are very, very busy tissues. A tremendous amount of your ATP reserves, of course. And so, again, we don't call these now. When we start to get into the electronical aspect, if that's the right word, the electrical aspect. We don't use the term end plate potentials are specific to the neuromuscular junction. And end plate potentials means it's going only one direction toward threshold. And if it reaches, we have an action potential. Here it's different. The action potential is all or none, but we have signals that basically is a combination of graded potentials. Some say fire, some say don't fire. It's a very simple system. Like I'm giving you information, you're making a decision whether to note it, write it down, to ignore me, to text somebody. Lord only knows what you're watching behind that screen. I know. I'll give you see what I'm saying. Your decision whether to do this, do that, or do that is all this complexity that comes on. Either do this or don't. Those are graded potentials. And it's very simple. It's either fire or don't fire. And you'll be amazed. And unlike the neuromuscular junction, where there's only one neurotransmitter, most nerves have more than one, depending on the signal frequency. And some say fire and some say don't fire. And some change when they're damaged, where then all of a sudden they perennially say fire. That's why people with strokes have spasm. They become spastic because all of a sudden they start to release tons of a neurotransmitter comes from glutamic acid called glutamate, which is always excitatory. Fascinating. So in the brain, finer dendrites are highly specialized. They have, ends that are, and it's a really good illustration. And all of that information coming in, and this would be the start of the axon, which I'm tracing with a cursor. That's the so-called axon hillock. So those signals come in, and if it is sufficient to get the threshold, we're not going to do the electrical aspects of it today. Basically, it reaches here, and basically that's where the threshold occurs, and then that signal goes in. When it goes all the way out to how many other little endings that axon has. So that's the axon hillock, this conical area. There's oddballs. Okay, you're going to see there's ones that don't necessarily have an axon. And imagine how long this thinner than a hair can be. We saw this with a muscle fiber. It could be a foot long. These can be a meter long. Like your brain telling your big toe to come up or push down. If you're 6 feet tall, 6'2", whatever, you can easily have a meter going down to the lumbosacral area and a meter going out to your lower extremity, to a nerve triggering the flexor hallucis longus, which would be the muscle that would be involved, or the extensor hallucis longus, or brevis. So we call them nerve fibers, okay? And a lot of times they'll have little collateral channels. They can modify the signal as it's going. It's like push it on the gas. Do I have to jam it on the gas pedal, or do you want to accelerate but slowly? That type of thing. So you can have multiple inputs or multiple outputs. The little terminals, you could call them buttons, the way it would be pronounced. Again, it's French, bouton. Love that. There's a great technique for sewing tendons called Brunel bouton. Sterling Bunnell was the father of modern extremity surgery, hand surgery. A lot of the stitching and sewing techniques are named after him. I did actually do this stuff. Don't look at me like that. You don't believe me. She's sure. Joey's skeptical. So, effectively, just dendrites receive, axons transmit. The impulses go along the membrane, as you might guess, because it's all about the membrane. And they secrete we've seen this movie before, neurotransmitters. They can excite or inhibit. Sometimes they do both. Lots of different conversations. The cell bodies, when we do a lot of reuptake of neurotransmitters, neurotransmitters get pulled in, okay, recycled, remade. And yes, if they're damaged, those axons, and thankfully, there's opportunities for repair. Some of them will repair themselves if the damage is not so severe. So the internal, it's all these neurofibrils. These are neurofibrillary core, sort of passageways that are probably analogous to microtubular-like structures within that nerve. And that's a transport system that, if it's signal only goes anterograde. Okay, so that's along the membrane, but it will take mitochondria, because you need a lot of them where you're going to release the neurotransmitters, additional cytoskeletal elements, membrane components, enzymes that are utilized for this process, either breakdown or various other properties. That's all going in one direction, from the cell body to wherever the terminus is. Retrograde, organelles can come back and be degraded. Neurotransmitters, signal molecules, can be recycled, remodified. Now the bad news. This is how viruses and certain toxins, most notoriously called neurotoxins, tetanus, botulism, rabies, polio. They hijack normal mechanisms. It's the interesting thing about this. So you have to be very understanding about this. Polio, rabies, herpes, which basically lays dormant in a dorsal root ganglia of any one of your sensory nerves, and it can be anywhere, and it can come back out at any time. I mean, ask Dr. Buxton about his shingles that he had. Oh, it was a number of years ago. Not good. So research underway using retrograde transport to treat genetic diseases, blah, 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 blah. I'm sure it is. So, I mean, anytime there's something that happens, particularly processes, we wonder how we can utilize them. It's like herpetologists, why they want effectively snake venom. Okay, some of them are neurotoxins, some of them are hemotoxins, so they can be utilized for a variety of different purposes. And so you take something like that, understanding the process, and then using it for some type of benefit. Maybe it can be destructive selectively to cancer cells, arbitrarily. Maybe we can take that toxin, that neurotoxin, and tag something with it that'll help. It's interesting, of the four, probably told you this, 4 snakes indigenous to the United States that are venomous. Rattlesnakes, copperheads, water moccasins, and the cottonmouth water moccasin is all one name. And the other guy is in the desert southwest. It's called coral snake. Looks like a little bracelet. The neurotoxins are the copperhead and the coral snake. And the other ones are hemotoxins. And if you really want to go back to 1955, there's a great movie about coral snake called We're No Angels. Some wonderful actors. It's a Christmas movie. 3 escapees from Devil's Island who go land in French Guiana on the mainland and help a very wonderful family to get their lives back in order. And really didn't like the way life was on the island, so they ended up going back to Devil's Island. At least they knew what to expect. Humphrey Bogart, Peter Ustinov. God, you guys, I have to educate you. I'll give you a couple more minutes. Myelin, protein and lipid, insulates. The more myelin, the faster, the thicker the nerve, the thicker the myelin, the faster the signal. Okay, the sheath surrounds it. If it's named, it's going to be myelinated. At the very end, when it gets to its target, it loses the myelin. We would see this in the extremity. We follow the nerve, and we got down when it was inserting into, let's say, a muscle in the thenar eminence here, or in the foot, here in the plantar area, we would see it begin to demyelinate. It was there. So non-myelinated don't have the sheath, and yes, it's slower, but typically they're already at their destination. So peripheral nervous system, Schwann cell. This is the jelly roll thing. One cell, one segment. Again, the outer collar, what they call perinuclear cytoplasm, fancy name. Okay, and that bulge has the nucleus and the side. Don't care. It's all about the membrane. So it's not so much the cytoplasm. The membrane is just this fatty, insulating material. No channels, no carriers, and where the accumulation of those channels, those voltage sensitive or voltage gated sodium and then potassium channels, that's right where they are. All those other active transport, the sodium potassium pumps, right there at those sheath gaps, at those nodes of Rhonda VA. So here it is, wrapping around. You can see, wrap, wrap, wrap. Here's the nucleus. That's typical of the myelin sheath. Different, however, as you saw before, when it's an oligodendrocyte in the nervous system. So you can see that's a lot of myelin. These are all those neurofibrils that are within it, different kinds of organelles, pigmented material, etc. 24,000 times. So that's an individual. I mean, that's what we're looking at. Same thing. The sheath gaps. And we often see when they talk about collaterals, this is where other nerves can enter on those gap sites and influence it. Here's a signal being sent, and all of a sudden, oh, Lou, you better go grab that. Whoa, wait a minute. Might be hot. That kind of thing. For us, it's like I'm walking and it's in the house, and all of a sudden the dog leaves me a surprise. Just ready to step. Whoa! It's okay. He's at CBW for the next 7-8 days. Can't bow out. He's on vacation. If we're on vacation, he's having a party. I bought treats. I bought food. He's with his buds. He's getting a bath. So, same illustration you saw before. Central nervous system, up to 60 axons. The sheath gaps are there. They don't have that outer collar thing. Okay, basically, it's very, very similar. And so the distinction, white matter, anything that's myelinated, tracks all the big passageways in the central nervous system or the named nerves peripherally. Gray matter, everything else. Synapses, non-myelinated areas, cell bodies. That's gray matter. Interesting, that's much heavier. Remember we talked about cerebral cortex being mass-wise. So we don't have nearly as much gray matter, but mass-wise, it's significant. And they just show you the same illustration. And we're almost done. You'll be happy to know you'll be done with me for a week. I'll record and send you something for fun. You can hear waves crashing in the background. So, typical nerves that you see in the central nervous system are almost always multipolar. Even the ones going out the neuromuscular junction. I mean, it has a lot of inputs and basically one long output. It's by far the most common one, the central nervous system. Bipolar, we talked about. When we talked about special senses, remember? Right? Anatomically speaking, bipolar is just modifying the signal that we talked about before, right? Did we do that sensory? We did not do that yet. We did it in anatomy.
In the eye.
In the eye. We did that in the eye and the ear anatomically. We'll talk about it physiologically soon. So remember, you had the actual photoreceptor, then a bipolar, then a ganglionic cell. You remember that trio? That was a test question about that, as I recall. So you see those special cells, and they're really just modifying or transducing the signal from the electrical impulse by, let's say, the photo to a signal that your brain can now interpret. And then unipolar, basically those are all sensory sometimes. And that's just the way they look diagrammatically. Not a whole lot here. This is common. We see a lot. The cerebellum has more synapses than any place else. That's called a Purkinje cell. That's the way you pronounce that. These are some of the pyramidal cells. Again, those are the big motor cells that are the main way that we tell our muscles to work. You can see the bipolars. etc., and sensory motor, mostly interneurons, the vast majority. Not a whole lot else to say. Goodbye. See you guys in a week. And I'll be honest, hopefully I will be back and be able to do this exactly. If I can't, we'll deal with it regardless. But I'm hopeful to have enough. I probably won't be back Monday morning, but Monday afternoon, I'll do my 3 lectures and then we'll do lab because we need to catch up. Thank you. Thanks for letting me not bother you in lab today. I know it's just,